Hello, everybody, and welcome. I'm Abby Chu, the Chief Scientific Officer of Precisionary Instruments. I'm delighted to introduce today's speaker, Sampada Tamhankar, a PhD candidate in the Department of Drug Discovery and Development at Auburn University. Sampada here is a member of Dr. Rajesh Amin's laboratory, a research group dedicated to developing innovative small molecule therapeutics for neurodegenerative diseases such as Alzheimer's and frontotemporal dementia. Sampada's work focuses on understanding CNS remyelination in FTD using a novel LXR agonist, AU403. As part of this research, she has developed deep expertise in mouse organotypic brain slice preparation using the Precisionary Compresstome vibratome, an approach that allows investigators to study myelin repair mechanisms with remarkable clarity and reproducibility. Today, she'll be sharing the methodology behind these organotypic slice cultures, including brain preparation, slicing workflow, and the experimental outcomes you can expect when applying this platform to remyelination studies. We're thrilled to have her here with us, so please join me in welcoming Sampada Tamhankar.
Thank you so much, Abby, for the wonderful introduction. So today I'll be talking on mouse organotypic brain slices, a powerful platform to investigate remyelination mechanisms in frontotemporal dementia. To begin with, first let's understand what's frontotemporal dementia. Frontotemporal dementia, or FTD, is a devastating group of degenerative disorders attacking the frontal and the temporal lobes of the brain, robbing the individuals of their personalities, behavior, and connections through language. It's an early onset disease, often before the age of 60, with symptoms such as impulsivity, apathy, which is loss of interest in activities, and communication difficulty. Military veterans represent a large population of the FTD patients. Public awareness has increased, especially following actor Bruce Willis's diagnosis, yet FTD remains widely under-recognized and frequently misdiagnosed. Currently, there is no FDA-approved treatment that exists, underscoring the urgent need to develop disease-relevant models and remyelination-focused therapies. Various collaborative research and increased advocacy are critical to understand and advance the care for those affected with FTD. Statistically, around 50 to 60,000 people were estimated by the National Organization for Rare Disorders in 2019 to be affected with FTD all over the state. But this number has now risen to 3 to 4 million people worldwide affected with this rare devastating disease. The most common form of dementia is this frontotemporal dementia, which occurs between the age of 45 to 64, and it's often misdiagnosed as Alzheimer's disease, depression, Parkinson, or other psychiatric conditions. This highlights the urgent need for creating novel approaches and therapeutics for treatment of frontotemporal dementia. Now, there are different models available for different forms of dementia, but currently there is no specific model available for FTD. So to study this disease, there are approaches such as 2D cell culture model, which includes induced pluripotent stem cells to differentiate into cells such as neurons and glial cells, including astrocytes, oligodendrocytes, and microglia. But as it suggests, the 2D cell culture has its own limitations. So how can we study FTD better? That is where comes in our involvement in these organotypic brain slices. These organotypic brain slices act as an ex vivo system, which provides a different approach other than the in vitro system and helps in developing these approaches for understanding this aspect of remyelination in FTD. But people might wonder, is an in vivo system one of the best systems? And why do we need better preclinical models? Now, let me tell you, mouse models in drug development reflect the best human form of neurobiology and disease mechanisms. But these direct in vivo testings are expensive, they are slow, and most importantly, they're ethically limited. This is where we need a middle ground where early screening can be done using an ex vivo model that retains the brain architecture and also overcomes the limitations of 2D cell culture. To give a brief of drug development and biochemical assays, I have an image here which shows that the initial steps involved in vivo or in silico modeling, which helps in computationally prediction of IC50 values, which is done in silico predictions. Post that, we have these biochemical and biophysical assays, which are high throughput assays, such as luciferase, TSA, and surface plasmon resonance, which help us in allowing to find the lead compound. It's also called as the lead compound identification step. This step is then followed by in vitro analysis. In vitro analysis is one of the strongest part in this step, which allows us to identify the best lead compound, its efficacy, and then goes on into in vivo testing. Now, the one of the problems which goes from in vitro to in vivo is that we need to identify the best drug possible, its efficacy and its toxicity. And most of these experiments rely heavily on in vitro testing as there is no permit available for in vivo unless you have confirmatory of the toxicity of your drugs. So now this is where we think the best way to evaluate our drugs other than just in vitro testings is where we will be having organotypic slices, because the limitations to these 2D cell culture or just cell culture is that these are four cellular organizations that hold polarity. They have very less or restricted cell-cell interaction, which prevents them from interacting with one cell to another. So this is the need why we need a 3D extracellular matrix or an organotypic slice where the different cells of your brain can interact with each other and create a physiological environment as that of the brain. So that's where organotypic slices offer a very better model compared to in vitro and before in vivo testing. We in Amin Lab use organotypic slices to evaluate the remyelination potential of a novel LXR liver X beta agonist drug AU403. These slices maintain native brain architecture, myelinated axons, neurons, astrocytes, and microglia. Thus, they allow accurate assessment of drug penetration and tissue level effects. These slices also provide a realistic platform for measuring myelin associated proteins, which focus on myelin restoration, then measuring the neuronal or dendritic complexities, help us in easy assessment of drug toxicity and viability, and lastly, structural remodeling over time. Overall, organotypic slices form a critical bridge between the 2D culture experiments and in vivo mouse studies. But before going ahead, let me just give a brief of what this aspect of myelination is. Myelin is a fatty protective layer that surrounds the axon of a neuron. After surrounding the axon, it helps in smooth transmission of impulses. Myelin is made up of lipids and proteins. As you can see on your right, it's made up of proteins such as PLP, which is proteolipid, MBP, which is myelin basic protein, and other lipids such as phospholipid, glycolipids, and cholesterol. Now, during disease states such as frontotemporal dementia, this myelin starts degrading. Myelin, which is produced by specialized cells called as oligodendrocytes, these oligodendrocytes start to degrade and die. Once they start degrading, they stop the production of myelin. Another mechanism by which we get remyelination is that these oligodendrocytes or precursor form of oligodendrocytes, which need to be matured to form mature oligodendrocytes that produce myelin, the differentiation gets hindered. And hence there's decrease in the number of mature form, which then causes demyelination. Now to cure this, this is where our drug comes in. The LXR compound AU403 works in two different mechanisms. First, it promotes the myelination by differentiating precursor oligodendrocyte or oligodendrocyte progenitor cells. It helps it in differentiating into mature oligodendrocyte, which then produces myelin. And the second mechanism acts by preventing the damage to the mature oligodendrocytes. This technique has extremely helped us in screening a number of compounds through high throughput screening, where we have tested a number of nuclear receptors to identify which drug could help us support remyelination better in case of FTD. To study this aspect of FTD, we isolate brain slices from a C57 mouse, and then to create the similar condition as that of an FTD brain, we treat it with a demyelinating agent called as cuprizone. Cuprizone, you can think of it, it's like a chelating agent which helps in reduction of the myelin by first preventing the differentiation of precursor cells to mature form, and second, by damaging and destroying the mature form of oligodendrocytes. So this is how we have utilized, how we screen our compounds. In further slides, I'll be also discussing our treatment plan and how we utilize this method. So the advantages of organotypic slices are, according to the NIH policies and IACUC policies, it helps us in replacing, reducing, and refining the use of mouse models. First, it helps us in replacing animal models as we can use lesser number of brain slices as compared to entire mouse. It helps us in reducing the number or counts of these mouse as one single brain can provide us with 15 to 16 slices which act as individual models themselves. So that reduces the number of mouse we have to use in vivo analysis. And lastly, refining. So we all know that mouse can serve as the best model for any disease state, but that also comes with its own limitations where the mouse has to suffer because of the diseases. This, since organotypic slices is an endpoint analysis or an endpoint procedure, we do not have any mouse suffering with any form of anomalies. It is just the slices which are involved in the treatment. Which now brings us to the features of our organotypic slices. To begin with, these are 3D tissue growth facilitating slices that creates an environment very similar to that of a brain environment or brain architecture. These slices that we use in our lab are grown on a transwell insert. This insert allows the organotypic slice to respire from both the sides of the organotypic slices. This in turn allows the gas liquid interface from the slice towards the medium. Besides that, these inserts are also flexible in shapes and sizes, so we can use them in a 6-well or a 24-well plate, which then facilitates for the growth using different materials. And also, these inserts can be used for different culture dishes. This is the insert that we use in our experiment. I performed the experiment using a 24-well plate, and we use these inserts from Sigma Mellipore, where these inserts allow respiration through the PTFE membrane, which is also a hydrophilic and a sterile membrane that allows organotypic slices to respire and breathe and also grow for over 10 days. Besides these, we also use other inserts that are shown in these images. These are 24-well hanging cell culture inserts, which are critical when you don't want the inserts to stick to the base of your cell culture plates. Similarly, in case of experiments, when you want to run replicates and you need same drug treatment, you need bigger wells. So we also have these 6-well standing inserts that can provide to accommodate more than 4 to 5 organotypic slices in one cell culture. Following this, as like every journey has its own roads and bumps, even our journey of organotypic slices had its own bumps in the road. To begin with the first one, the agar scaffold. So while slicing these brain slices, we have to mold them into a special form of agar. Initially, we tried using 5%, 2%, 1% regular temperature agar, but that failed us in creating even slices. And also the heat produced by the agar caused damage to the cells of brain. So that's when we shifted to this specific low gelling temperature agar, which is from Thermoscientific, which solidifies at 36 degrees Celsius and then it stays solid even at room temperature. This helps us to create a scaffold which is not of higher temperature and prevents the damage to our brain cells. Second, proper buffer to ensure cell viability. In our initial experiments, we proposed to use media which consisted of glutamax and different nutrients which are critical for formation of and differentiation of neurons, but we failed to attain the ACSF condition, which is artificial cerebrospinal fluid. So, followed by this, we did a couple of trial and errors using the thermoscientific protocol, which is a STAR protocol, and we made our two different media. One is for brain slicing media, and second is for brain culturing media, and that helped us to overcome this bump. The next one would be membrane placement and immunostaining thickness based. So these problems were then resolved using multiple combinations of antibodies or different proportions, which helped us to understand the exact immunostaining properties for these organotypic slices. And the last two parameters, which is sterility and section thickness. These were two of the major problems that or hurdles that we were facing. For it concerned as sterility, our initial vibratomes that we used created chatter marks and we were not able to sterilize the instrument itself because it was completely screwed onto each other. And the second part was the section thickness. We couldn't attain a smoother section thickness because of the chatter marks. But then this is where the precisionary vibrating microtome came into rescue. Since this microtome can be disassembled easily, we could clean it and sterilize all the individual components of this device. It was consistent in even slicing and also compatible with low melting agar. It left no chatter marks and was easy to sterilize, which also reduced the mechanical stress and then supported variable slice thickness. This is the setup that we have in our lab. We have set up the whole as experiment of organotypic slices in a BSL hood, where beginning from the left, these are the instruments that we use for dissection. We have specialized paint brushes, which we have gotten just for isolating and collecting the brain slices from the tank. Then followed by that, we have different buffers available, which we oxygenate inside the hood to maintain the respiration of these organotypic slices. Now moving on to the best part, our experiment, organotypic brain slices protocol to experiments. Initially, we begin with mouse brain isolation. After anesthesia, we process with brain removal and then rinse this brain in 1x ice cold PBS. After rinsing it twice with ice cold PBS, we then transfer it on icebox and bring it to our BSL biosafety hood and then proceed with desired method of sectioning. Now, what I mean by desired method of sectioning is that there are different neuroanatomical directions of plane. And then depending on your research or depending on what you're looking at in the brain, you can either do a frontal or a coronal sectioning, a horizontal or an axial sectioning, and lastly, a sagittal sectioning. So for our experiment, since we are looking at the overall brain diversity and density, and we also need to focus on the hippocampus and frontal region, we focused on the coronal sectioning. In our process, we first isolated a brain from a 3-month-old C57 mouse, and we immediately transferred it to a petri dish with chilled slicing medium. After transferring it, we then mounted it in a low melting agar block using this cell culture slicing medium. Then, with this agar block, we mounted it onto the vibratome. So this is the region where we mount this agar block, and then we section it horizontally or coronally at 250 micron thickness using a sliding vibratome in an oxygenated environment. It is very important to make sure that all the media and slicing medium we use is well oxygenated so as to prevent any sort of contamination and damage to all the cells in the brain slices. Post slicing, we transfer these intact slices onto a well insert. They place into an insert which is into a 24 well plate containing the cell culture medium. So this is where we change from cell slicing medium to the slice culture medium. And post culturing, we maintain the slices at 35 degrees Celsius in a fully humidified 5% CO2 atmosphere, replacing the medium every second day. Now, as I mentioned before, to study this model of frontal temporal dementia, since we have isolated slices from a C57 mouse, we need to create environment as that of the frontal temporal dementia patients. To create that environment, we use this drug called as cuprizone. Cuprizone helps us in removal of myelin sheath, which is a key pathology in case of FTD, which is also called as demyelination. After 3 days of treatment with cuprizone, which causes around 80 to 90% removal of myelin, we then start treatment with our drug called as AU403. In our drug treatment, we have a plan of 7 to 10 days of drug treatment, where after every 3 days, we isolate the slice and look at the expression and the protein expression of these myelin associated genes. Lastly, after isolation of these slices, we then follow by our immunostaining protocol. It is as follows. The first step is fixation with 4% paraformaldehyde at 4 degrees for 24 hours. Followed by that, we wash with 1x PBS for 2 times and followed by the step of permeabilization. Permeabilization allows the antibodies to go inside through the slices, and this permeabilization is done using 0.1% Triton X100 at 4 degrees Celsius for 24 hours. Followed by that, we block with 5% goat serum and then add primary antibody, which goes on for 48 hours at 4 degrees. Followed by primary antibody. If it is not tagged with a fluorescence fluorophore, we then move on to a secondary antibody, which is incubated again for 24 hours at 4 degrees Celsius. And the last step in our immunostaining protocol is the DAPI staining for nuclear localization. Lastly, we mount these slices on a slide with anti-fade gold media, which prevents us the loss of fluorescence. And lastly, this fluorescence is measured using a fluorescence microscope. Now comes my favorite and exciting part, which is the results. So, our first results, we highlight the myelin basic protein, which is a key component and a biomarker of myelin sheaths inside the brain. As we can observe in the first panel where we have treated with cuprizone, which is demyelinating agent, we barely see any myelin basic protein, which is shown in green fluorescence. This is then reversed with treatment of our drug AU403. The second panel is a control which was treated with 403 since day one. But then the third panel was first demyelinated with cuprizone for 3 days and then treated with AU403 for another 7 days, which helped us in reversal of the demyelination effects. Similar results was also observed with another form of marker, which is called as proteolipid 1. This is another form of myelin mature marker, which helps us in understanding the remyelination properties. Besides remyelination, we have also used organotypic slices in our other findings, which is the rapid Golgi-Cox staining. This rapid Golgi-Cox staining is used to quantify dendritic spines, their morphology and density around the neuron. These are small projections around the axon of neuron, which are involved in transmitting impulses from one synapse to another. Now, depending upon their shapes and sizes, they are classified either into neurodegeneration, where you see thin and filopodia-like spines, or growth, where you see thin but stubby spines, and lastly, the spines responsible for transmission, such as mushroom and branch. Now, as it suggests, when you have a healthy neuron, you expect to see more level of transmission spines, such as mushroom and branch. And when there's neurodegeneration, you expect to see more level of filopodia and thin spines. So when we did our experiments, we did these organotypic slices, and we then treated them with this rapid Golgi-Cox staining. After staining, and in comparison with a pre and post drug treatment, we observed that the C57 mouse has higher number of mushroom spines, which is critical for transmission. Then comes our 5X FAD mouse, which is the transgenic Alzheimer's disease and a front ADRD mouse, which shows reduction in these mushroom spines, which suggests that there is less number of transmission spines, but interestingly, there is higher number of neurodegeneration spines, which is filopodia, stubby, and thin. But then 1 month treatment with the AU403 showed reversal and caused increase in the mushroom-shaped spines. This was one of the best findings to show that it just does not improve myelination, but it also improves the synaptic plasticity of these neurons. Now, moving ahead, our future work focuses on spatial genomics. Spatial transcriptomics maps gene expression across intact brain tissues. So, using these organotypic slices, we plan to send it to spatial genomics, who will help us identify different drug treated regions and identify which myelin repair pathway is involved. We plan to look at integrating spatial genomics with different drug exposure, which will help us reveal cell type specific signaling networks that are activated by our Alexa drug, which will help us pinpoint the molecular drivers of remyelination. Lastly, I would like to acknowledge and thank Harrison College of Pharmacy and Department of Drug Discovery and Development for providing a diverse research environment that allows us burgeoning independent scientists the necessary tools to succeed. I would also like to shout out and give a big thank to my advisor, Dr. Rajesh Amin, for his constant guidance and support. Also, my dear lab mates who've been there with me. A big thank to NIH and AHA, American Heart Association, for their constant support and funding. And lastly, thank you so much, everyone, for listening so patiently.
Thank you very much. I wanted to open up, and I didn't see any comments. But sometimes our audience can be shy, and we'll have follow-up questions. So what I like to do, and still open it up to see if anybody has any questions. I see Dr. Brian Christie is here. Hi. Please feel free to unmute and ask questions. And if not, I would love to wrap it up and thank our wonderful speaker today. So Sampada and Professor Amin, thank you very much for your time. And for our audience, feel free to write to me to be put in touch with them if you would like to ask any questions outside of this webinar. Thank you very much, everybody.
Thank you.